Okay, I would say that we get started with the second session. This is a more scientific session now. And I will introduce Sebastian Burkhardt from Heidelberg coming, who will present his work on this 15q genotype.
Hi. So it's a pleasure to present you our efforts regarding an in-depth analysis of the Dup15q genotype. The rationale for this is quite obvious. The symptoms of Dup15q syndrome are highly variable and without a clear genotype-phenotype correlation. And in our work, we try to find differences in the genetic variation who might explain the differences in phenotype or in the clinical symptoms. And for that, we started by generating a literature-derived cohort. So we started by reviewing 5 established research databases, and from these databases we excluded all publications with any patient-level genotype data, and this patient-level genotype data was then extracted. And with that we were able to collect all published cases with individual-level genotype data, which is about 421 cases. So this approach also faces some challenges, and the major challenge is the limited data availability. If you look at this bar plot here, you can see that in about 30% of the cases, there's no information on which genetic test is used during genotyping. And if you look at the bar plots here on the right, you can see that parental origin is also only known in about 20% of the cases. So if the chromosome is really maternal derived, we don't know it. And there's also some highly limited data, especially regarding the nucleotide level of the breakpoints involved and also of the size of the copy number gain. So this is really highly limited in the current literature. So now you have a short impression about this data set. You already heard something about this data set, so we also have an increasing number of individuals from our registry, currently 93 cases. In these bar plots, you probably recognize part of this figure. You can see the percentage of cases who received different diagnostic technologies. Here on the left side, you can see the data from the literature-derived cohort. Here on the right side, you can see the data from our registry. What you might see is that more, I call it, traditional diagnostic tests like karyotyping and FISH diagnostic are the dominant diagnostic approaches in characterizing these the Dup15q genotype. But these technologies are low resolution. If you look here in our registry data, we have an increasing number of individuals who received array diagnostic or even exome or genome sequencing, which gives us a little bit higher resolution of the involved genotype. Again, interestingly, the number of individuals who received an methylation-specific MLPA, which is a test to really validate the maternal derivation of the extra genetic material, is only about 10%. So often it's just assumed that this extra genetic material is really maternal derived or maternal imprinted. if we look at the different genetic subtypes within the both cohorts, we can see that a majority of cases have an isodecentric chromosome 15, so that's the green ones. In about 20%, we can see the interstitial variants, so this is also in line with the estimates from the literature. But as our data is derived from routine genetic testing, in some cases or a number of cases, the really location of the extra genetic material, if it's interstitial or if it's an extra chromosome, is just not determined. So in the next slide, we will focus especially on the EDIC-15. We will start here with the general structure of a EDIC-15. So we are going from left to right. So you got here a P-terminal region. Then you got a centromeric region in red. Then you have genetic material of the 15q arm with different breakpoints. And then you have again genetic material of the 15q arm, again with 5 different breakpoints. Again, a centromere, and then a P-terminal region. The structure, especially regarding these breakpoints or the breakpoint combinations within individual EDIC-15 is highly variable. So that's why we analyze the frequency of different breakpoint combinations of this EDIC-15. And that's what you can see here in this dot plot. So here are breakpoints. And we were able to identify 2 main clusters of EDIC-15. One is a breakpoint 3 combination, which results in a symmetric EDIC-15. And another one is a breakpoint 5 combination, which results in a more asymmetric EDIC-15. And this frequency is also what we can see in our registry data. So we have a substantial amount of individuals who have a more symmetric EDIC-15. We have a substantial amount of individuals who have an asymmetric EDIC-15. But we have also some cases where the really architecture of the EDIC-15 is just not determined. So maybe to conclude this, now we have 2 large data sets with individual patient-level genotype data available, one literature-derived and one from our registry. We were able to identify 2 main clusters of EDIC-15 formation, one symmetric one and one asymmetric one. But there are also some significant gaps within the molecular characterization of the EDIC-15, especially regarding the imprinting or the high-resolution structural information of the EDIC-15, and also the nucleotide level breakpoint information involved in EDIC formation. And now I will give you a short outlook on what we are planning or what we are currently doing to address these challenges. We are doing ultra long read sequencing. For those of you who don't know what ultra long read sequencing is, it's a novel genetic test. It's a novel cutting edge technology. And with that test, you can simultaneously measure the methylation. You can measure or you can identify structural variants. And you can also get high resolution sequence data from this EDIC-15 chromosome. And I think with this data, we are able to really address the question of structure within this EDIC-15. We have already collected more than 50 blood probes, and currently I think 10 probes are in sequencing. So we are really looking forward to see this data, and I would be happy to share this data with you in the next Dup15Q symposium. And thank you very much.
Okay, thank you, Sebastian. I think all these questions regarding this testing will be discussed afterwards. One point, there's a list going through all people who is interested to join us on our European collaboration on Dup15Q. Please write your email down, and so you will be in our group. The next presentation comes from Julian Nevado. He's a Spain researcher, but he is not able to join us today, so he has recorded his presentation, and I hope that will be work.
Hello everyone, my name is Julian Nevado and I'll talk today about our project with DUP15Q syndrome in the Hospital University of La Paz in Madrid. This is our workflow right now. It is the project is pretty much down in many of the aspects. We made patient evaluation, validation of results, karyotype-phenotype and molecular analysis for many of the patients. We are starting to get the comparison and analysis of the results for methylation. Current situation of the project starting with 74 individuals in 2023, mainly from different regions of Spain and Latin America, mainly Argentina, Chile, and Uruguay. And after grading the results of the patient, repeating many of that results with SNP array, we finally get 41 individuals with the DUP15Q syndrome, and after that, right now in 2025, we have 2019 patients more. They're given a total of 62 patients, 35 males and 27 females. Methodology is a prospective observational cohort study with patients contacted through the foundation of invdup15Q in Madrid and the University La Paz University Hospital. We get a medical record and several questionnaires from the parents and after confirmation, the diagnosis. The main aims of this project it was the creation of analysis of registry database and finally to establish or rule out a possible genotype-phenotype correlations in this syndrome. We started with the visualization and extrapolation of data from medical reports and questionnaires, getting to an Excel for working as a registry database, including almost 60 variables. And we have to highlight one of them, the creation of GFAP, the Global Functional Assessment of the Patient. It's a functional score that we describe and validate statistically to get comparison between different patients. This global functional assessment of the patient is getting from different HPO weighted symptoms and terms in the syndrome, and is based on our clinical expertise regarding aspects of morbidity, development delay, dysmorphic feature, language anomalies, and behavior anomalies. All of them give you as individual in a score that give you a multi-construct after that. Basically, the characteristic of the cohort, and this is a descriptive statistics, most individuals were in the pediatric age between 0 and 12 years, around 78%, and the averages for median age at evaluation were almost 9 years and 7 years respectively. Most of them, they give a birth at terminus, and there is no significant alteration in weight, height, or head circumference, with most of them remain around P50. We have similar numbers of males and females individuals, and the presence of the marker is predominant around 78. We have many other data, but pretty much they are very similar that were previously described for the syndrome. Samples to work. Basically, we have in 50 individuals preliminary data. It came from most of them, 78%, wearing a possible marker the isocentric marker chromosome, extra chromosome. How is the results from the arrays? We have duplication, triplications. We also more complex rearrangement, duplication, or duplication. And some of the cases, in two of them, are very rare results, including partial exosomes or even autosomies. The minimal SRO, or small region of overlapping, trying to look for which genes are involved in our cohort in this small region overlapped. And as such, the genes are in highlights in the box, and UBA3A, GABA3, GABA3, 5, OCA2, R2. As previously it was described, involved in. We also make different clusters by principal components and using different variables like morbidity, development delay, language impairment, behavioral problems, dimorphic traits, or methylation pattern as nuclear features of the syndrome. And we get three independent clusters we call mild, severe, or intermediate. It depends of the presence of different degrees of a marker and different degrees of epilepsy and HD. And development variable, we are almost explain 80% of the cases where our variable GFP is the most valuable variable to explain or to classify these individuals in the three classes. We also starting to ask about is there any relationship between core functional variable that represents the marker, or question as is there any relationship between the size of the duplication and severity? We can say that the marker chromosome is associated with larger duplication size, a high number of genes, and a more severe clinical burden, particularly in the presence of epilepsy. Moreover, GFAP is significantly increased in epilepsy with a marker, suggesting a relevant functional role in the pathogenesis. Regarding the marker. Regarding the epilepsy, we have that epilepsy is significantly associated with the presence of the marker chromosome. Instead, patients with the marker have a higher incidence of epilepsy. Also, GFAP is a marker of severity, where it's elevated in patients with epilepsy and the marker, suggesting greater functional impairment and potentially worse neurological prognosis. The marker's effect is not solely due to larger duplication or more genes. This may suggest that the clinical impact depends not only on the replication size, but on how it modulates gene expression. That means that we check the methylation pattern in the presence of the marker, and you see that the presence of the marker, most of them cases are hypermethylated, and no marker is rare in the methylation alteration. So the presence of the marker chromosome is associated with significantly larger duplication and higher frequency of hypermethylation. In contrast, patient without the marker shows smaller duplication and virtually no hypermethylation. This pattern suggests that both sides of the duplication and the structure, presence of the marker, may suggest an impact on epigenetic regulation, specifically and in hypermethylation. That is our supposed hypothesis regarding a possible hypersignature. Regarding that, epigenetic and imprinting, UBA3A is expressed only from the maternal allele in the brain, and maternal duplication leads to the UBA3EA overexpression. So, in understanding imprinting improved diagnosis in the syndrome, maybe, and epigenetic studies maybe will open new avenues for the research. That's that we are studying a possible existence of episignature in the syndrome, and we are evaluating already 15 patients of that. So that is the most recent focus in this project. Thank you for your attention, and maybe we can have some political question. Thank you.
So the applause is for our technicians. Thank you very much for making this possible. Okay, I think it's a interesting point and very important point. We thought about this also. Okay, the next talk I'm looking forward is Karen. On my list is Genotype-phenotype association in the Dup15Q cohort, and I'm very excited what you will present us.
Okay, this is where we stayed with the talk of Marie-Claire. We started this registration with the analysis of it with 37 patients, and I'm going to tell you about the results that we found. As I think, because I'm the third or fourth with this natural history overview, there's going to be a lot of repetition, but some things are a little different. So that's where the discussion, I think, is most interesting. In our cohort, there was also a quite comparable sex. Thank you. The age of the patients that we analyzed was had a mean of 12 years, but with a wide range, with the youngest 2 years old and the oldest 28. Well, this is new. We saw actually that the children had a lower height, like -1, -2 lower standard deviation score, with not significant, but a difference between the interstitial and the edic group. And this was also counting for the weight, which was the significant difference. So the edic children had a lower weight. Most of the children live at home, but some already live partially or completely in an institution. And there was some difference that the interstitial children mostly living at home, but that, well, we haven't related that to the age yet, so that's to be continued. And unfortunately, we lost one patient quite recently, actually. In terms of the genotype specification, we have 16 children with an interstitial duplication and 21 children with an edic, and that's actually more like comparable to each other in comparing to the earlier talks. I don't know why. And most of them, or actually all patients had the duplication between at least the BP2 and BP3. So we didn't have duplication children with only BP3 or BP2, BP4 or 5. So that's an interesting thing, because we always think that the UBA3A is one of the, well, actually the main gene that is causing the phenotype. So that's interesting for later research, I think. We have 6 families, so then there are like 3 families that have 2 siblings with Dup15Q, but we also have like 2 families where the mother is also having the same duplication, and they're all interstitial, as we can imagine. And in terms of parent of origin, we know that, well, most of the children have a maternally derived duplication, but there's one who has a paternal, and he's also showing the signs of the duplication 15q. So that's interesting. We have 9 children that we do not have this information about yet, because they are already earlier diagnosed and only with an NLPA, and parents weren't very interested, or there was no child wish. So we've have contact with our lab now, and these 9 patients are going to be additional analyzed to see where, well, what the parent of origin is. So that's interesting, but that's also to be continued. And the size of the duplication is, as we've seen earlier, the interstitial have a smaller duplication size than the edic children. There was a long diagnosis odyssey, as we've seen in earlier talks, and, well, not surprisingly, the interstitial duplication children had a longer odyssey than the edic children, because they were more mildly affected. So we hope that with our collaboration, with more efficacy and awareness of this syndrome, we can shorten this. The leading point for diagnostics was most of the time like a motor delay, behavioral problems, generally. Actually, I think if you sort of summarize this pie, you can see that they're all developmental questions, like either speech language or motor, or more like a general developmental delay. The behavioral problems come later. So this. And we also asked the parents, what's your actually current concern mostly? And I think we've already heard them. Behavior is one, followed closely by epilepsy. And I think this is where our main goal lies in the future years. We used the clinical global impression scale, and actually there was not a lot of difference between the EDIC and the interstitial children in the answers. You see that like motor function, the ability to take care of yourself for at some level, and also behavior again and sleep. Epilepsy, 1/3 of the patients have developed epilepsy, and as you can see, the EDIC children have a higher prevalence of epilepsy than the interstitial children. And actually, for me, what was most striking thing is that I was sort of raised with the idea if you have a syndrome with a risk of epilepsy, it's sort of you have to debut in the first years. But here we see that you can also develop this at a really older age, like also above the age of 10. And we were quite impressed by the destructive effect that it had, that children had real regression signs and also did not catch up once the epilepsy was treated. In terms of severity, the EDIC children also have a more severe type of epilepsy, and you can see, for example, as the number of anti-seizure medication that is needed, the type of seizures that's diverse, but mostly the tonic-clonic seizures and the absences. That was not very new. Behavioral problems. 2/3 of the parents describe behavioral problems with almost half of them persisting. Hyperactivity, you have to be on all the time because they're everywhere. And the screaming, that's, I think for you quite recognizable. We also asked what are triggers, what triggers this behavioral, and I think that the last two are really important because when you cannot express what you want or what you don't want, you only have scream for an expression. And the overstimulation, I think it's quite fascinating that the children are on the one side very sensory seeking, and on the other side are so sensitive to stimuli. And I'll come back to that later. But the other thing is the autism. We ask, is there already an official diagnosis? If it's not, do you have suspicion? Well, there were no signs of autism in 1/3 of the group, which I sort of surprised because for me it's like all the children have autism. And there was 40% of the children already had an official diagnosis of autism. And the in-between group we provided with an additional questionnaire, which can sort of seek out if there's signs of autism. And 60% of the answered questions are giving signs of autism. Coming back to the stimuli, hair blower, vacuum cleaner, the thing that you blow in the autumn, the leaves, horrible for children. But also touch, light, smells. But the auditive it sort of springs out. Sleep, a lot of sleep problems, but it sort of seems that they're diminishing over time. So 50% is still a lot, but it's not the original 83% with sleep problems. And it's both settling to sleep, waking through the night, and very early waking, like 3 o'clock. Well, let's begin the new day. And actually, we didn't see a lot of difference between the interstitial and the EDIC group. It's like in the whole group. These were things that parents reported that helped, that they tried to diminish the sleep problems. I want to be short with that. All interstitial children learn to walk. And I think about, what was it? 40% of the children with EDIC do not walk or need support or are completely wheelchair bound. And the hypermobility, which was discussed earlier. Speech and language is actually a difficult one because it's so diverse. And I think this is what we gained from the parent-reported questionnaire, but I think we all really need also the findings of our speech and language therapists to have a more objective overview of the communication capabilities. But again, a difference between the interstitial and the edict children. And also the echolalia were, I think, I forgot who, yeah, you told. And we also had this concept of the Gestalt communication, that there are a lot of words, but they're not completely meaningless. They seem to use them. Yeah. So this for us is this sort of new finding. And then again, the growth, smaller, again, the genotype difference. This is weight for height, because as a pediatrician, you always have to look at the proportion, not only the weight, because, well, they're also small, but even when they're corrected for their height, they're still have a low, yeah, they're tiny. Yeah, they're this tiny, very active. We also looked at the head circumference, and that was actually quite normal. So, well, at least it was not completely normal, but within the normal range. A lot of gastrointestinal problems, but also they sort of grow over it in time, but not the whole group. So a third persists in having reflux and constipation. And my personal opinion of experience is that it can cause a lot of additional behavioral problems. So even if you think with all your physical symptoms there's no constipation, just treat it and see if it goes better. And if it doesn't, well, you can stop, but it has a lot of positive effect when it's treated well. Potty training is an important issue for parents. More than half of them are not able, but we still don't give up. Tube feeding, there was actually only one patient who had persistent tube feeding, so that was not too bad. Future plans, they're very important. This is a cross-sectional analysis of our first 35 patients, but it's very important to have longitudinal follow-up. How is it going over time? Well, we're, as I said, we're specifying the genotype, so we know more about this importance of this epigenetic factor, maternally or paternally derived, and we have to analyze the first findings of our outcome measures that the doctor reported data to have more objective from our side, to have a whole view of our group. And of course, as Marie-Claire already mentioned, we're progressing now into our child brain lab, to either see the more like cognitive functioning, like the eye tracking and the EEG, and otherwise the more like the growth parameters, like the bone health and the body composition and energy expenditure, what's going on there. And we, of course, are very happy to be here to share our experiences and work together in these future plans. So if you want to find us, this is how we can find. I think the most important lessons for us were, well, the first one is not surprising, but it's confirming. The second one was surprising for us. And the third one, I think, is the most important one. So let's work together. This is our team.
Okay, thank you, Karen. I think we have time for one or two questions. Is there? And yeah.
On the theme lower height and lower weight in the children, were there any seen of them by an endocrinologist? And is their growth hormone a topic?
Yeah, that's a very good question. As a pediatrician, I can do that myself. But the thing is, they're shorter, but not too short in terms of that you are, well, we have all these protocols. If you're below this or below that, you do this endocrine investigations. So I don't think they're like, but we do now in the brain lab. We just take blood from all the patients if possible. To see what's the range, what are the trends in hormone levels, both growth hormone, but also the thyroid and things like that. I had one patient with a celiac disease, but he was really below this -2 standard deviation, so that's how I got that. So I think it's really important to address this endocrine function more now. Is it an endocrine factor? Is it like a higher metabolism? We don't know yet. But I'm more aware of it now. Now we've seen this in this analysis.
My question is for every researcher. I was wondering if it makes sense to make genome sequences, because I asked this to my neurologist. It was very doubtful, but I think it would be a good idea to evaluate any comorbidity other than DUP15Q. I don't know, what do you think about genome sequences?
The genome and then?
Then,
Oh, sorry, yeah. Well, it's always good to evaluate for yourself, is this completely an explanation of what I see, or is there, well, do we have to consider other things? And we see patients would have a second diagnosis, either genetic or like more pediatrician-wise. And in the Netherlands, we have sort of a new protocol now in the last 2 years that children with developmental problems, we start with whole genome sequence. Oh, well, in Rotterdam we start with whole genome sequencing. In the rest of the Netherlands, it's whole exome sequencing. But so it's already done at the beginning now. So if there are a second diagnosis, they come up, and it's with copy number variant analyzed. So we pick up the DUP15Q.
I thought it was really interesting to see your cohort versus the Italian cohort and ours, and it really shows the bias of the pickup.
The numbers,
right?
yeah.
The hospital that works stronger on neurology obviously has more severe epilepsy. And I think that shows again how important it is to have these very large
We need more
groups
numbers
to look at.
And not the coincidences, but to see that in our cohort we're not the only one with the older age of epilepsy. Like, oh, it's not a coincidence. It's probably a trend. Yeah, but there are differences too. So normal height with this previous talk, and then and so I think every hospital can have its own hobbies, or I don't know. But if we work together and we can use each other's experience, I think that's the brilliance of this network meeting.
Yes, and if we match all the data and put them together, we get a better overview. Okay, thank you, Karen.